Okay, so welcome to biotechnology. It is chapter 20 in your book. All right, let's do it without further ado. Do, 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 dotty, do, dotty, do, dotty, do. Okay, so just want to point out that your genetics project information has now been posted in a new topic called genetics project, because this is going to kind of follow you throughout the semester. So we're going to do a little now with the research paper that is going to be due in early February. So that's coming up. And I know some people have gotten a good start on it. And I know some people have not thought about it at all. All right. Do not wait until the last minute, because there are going to be questions that you have that you want to talk to me about, and you're just not going to have that chance if you do it too late. All right. So again, what you're going to be focusing on is looking at the proteins that are affected. So remember, our DNA is going to have a mutation or two or three, whatever the mutations are, that is going to cause a problem with the protein, right? Here are the proteins that you're going to focus on. And your job is to figure out how these protein problems, how these problem proteins actually cause the disease. All right. And for some of you, it's going to be very clear. And some of you, it's going to be not clear. And you're going to have to do some research. All right. We're going to talk about this when we talk about in class. So remind me to introduce impossible foods. So again, Mr. Anderson, I'll post these videos. He does a good job of explaining biotechnology. And we are going to start by looking at the DNA toolbox. All right. So there are kind of 5 main tools that we are going to use here. All right, so the first is about restriction enzymes. These act like little tiny scissors that will cut DNA. All right, so that allows us to take DNA out of one genome and put it into something else. All right, we're going to see complementary base pairing. Again, that occurs by hydrogen bonds. And DNA ligase, we've already talked about, that is the enzyme that acts as the molecular glue to put phosphodiester bonds back together on the backbone. So here we can cut DNA out of a genome, and now we can glue it back in to someplace else. Right? We're going to see that gel electrophoresis is a way that we can separate different pieces of DNA, and this is going to be based on their size. Right? So for example, if we had a circle piece of DNA and we cut here and here, we can then run it on a gel to separate this smaller piece away from the bigger piece. Okay, we're going to use a technique called polymerase chain reaction. And this is a process that allows us to make many, many, many different copies of the DNA. So for example, in this example, if we wanted to take this little chunk of DNA out, we could run it on a gel, we could isolate it, and then we could use PCR to make millions of copies of that DNA. And then we can actually do stuff with it. It's very hard to deal with very small amounts of DNA. All right, and then the last thing is DNA sequencing. This is a technique that allows us to kind of determine the sequence of base pairs in that DNA. All right, and that has become very powerful. All right, and we're going to also talk about gene cloning, which is kind of shown a little bit down there. All right, so we're going to start with restriction enzymes that cut the DNA. So restriction enzymes cut DNA at very specific sequences called restriction sites. And they often leave what is called a single-stranded sticky overhang. All right. So for example, if we look at this is the enzyme. So this is the enzyme, and this is the information that you would get like in a catalog if you were going to buy this enzyme. Right? So these enzymes were originally found in prokaryotes, so in bacteria, and we now use them in research labs. So here's an example called EcoR1. And the way it cuts is it shows us this little diagram. It cuts like this. All right. So that little red arrow shows where the little molecular scissors are going to cut. Right. And so, for example, if we have a piece of double-stranded DNA here, EcoR1 is going to kind of bind that DNA, search for the site, and then it is going to cut in that pattern. And so what that does is it leaves these kind of overhangs. All right. And if you don't do anything at all, these guys might then stick back together by hydrogen bonding. Right. However, we could also cut a different piece of DNA with the same enzyme. Right. And that would leave the same overhangs. And what that allows us to do is to put two pieces of DNA from different sources together with DNA ligase. All right. And so when we look a little more carefully, so we're going to look up here in this diagram. If we look a little more carefully, if you notice the 5' and the 3' end of DNA, remember they're anti-parallel. When EcoR1 here made our cut, so it's GAATTG, notice we are reading from the 5' end. We see GAATTC. Notice if we read from this end, we also see GAATTC, right? So they're kind of palindromic writing from the 5' end. So when it cuts, we are going to have the end here. We have a 5' end and a 5' end. So these are 5' sticky ends, all right? They're sticky because of those hydrogen bonding complementary base pairing, right? So here is some practice with that, which we will hopefully do in class. So again, when we're looking at this, we're always going to put a 5' and a 3' end. We're going to actually add the second piece of DNA, and we are going to look where it cuts. One thing I want to note is this one here. Notice that it is going to cut right in the middle. So this will leave what's called a blunt end cut. So instead of having those sticky ends, they're going to have no sticky ends. All right. So here's an example where we are going to cut DNA with a restriction enzyme and glue it back together with DNA ligase. All right. And that is going to create what's called recombinant DNA, the combining DNA from two different sources into the same DNA molecule. All right, so let's go through this. So the first thing we're going to do is to use restriction enzymes, which are the little molecular scissors, and it's going to cut the DNA. Notice here, the enzyme that we're using is still that EcoR1, and it's going to cut in this weird pattern, right? Right between the first GA, right? So what that's going to do is that, let's just draw our ends here. That is going to leave 5' sticky ends. And we're going to cut a different piece of DNA. So we're going to cut different DNA with same enzyme. So it's going to leave the same sticky ends. That DNA fragment from another source, from both sources, are going to be added together, and those sticky ends are going to be complementary. So therefore, you can use DNA ligase to seal that together. So now we have a piece of recombinant DNA. We have DNA from this green source, and we have DNA from this gray source. What we're going to see is that the DNA sequence of your choice, so an example of kind of doing this, kind of using these restriction enzymes and DNA ligase, is that we could take DNA from a sequence of our choice. So let's say we wanted to isolate the cystic fibrosis gene from humans. All right. And we wanted to put that into an organism of choice. Usually what we're going to be using is called a bacterial plasmid to help us do that. all right? And plasmids can be really confusing for students. So we're gonna go over what that is. So what is a bacterial plasmid? A bacterial plasmid is all it is a circular piece of double-stranded DNA distinct from the bacterial chromosomal DNA. So here, the plasmid is just a circle piece of DNA. And I want to be clear that this does occur in nature. These were determined, this is a real phenomenon. Bacteria often use these plasmids to help them gain some sort of selective advantage, right? So they can pick up DNA from their environment. Remember that? Okay. And do you remember what that's called when bacteria pick up DNA from their environment? It's called transformation, right? Okay, so the elements of a plasmid, what are on these little plasmids, right? So in a lab, we use plasmids all the time, and they have specific features. They have an origin of replication, which is kind of not shown here. So it's called an ORI, an origin of replication. And that will ensure that as the bacteria cell grows and divides, we are going to replicate not only the bacterial chromosome, but also the bacterial plasmid, right? It has a promoter. Remember, promoters are necessary in order to drive gene expression. And when I mean gene expression, I mean it will help. Actually, I'm not going to say it help. It's required to start transcription, right? Remember, of the making of RNA from the DNA, right? We have our target gene of interest, whatever we're studying. In this case, we are talking about studying the cystic fibrosis gene. And it has something really important called antibiotic resistance. So an antibiotic is something that will kill bacteria. All right, bacteria specifically, not viruses, bacteria, right? If we put an antibiotic resistance gene in there, then this plasmid will grow on that specific antibiotic. And that will allow for us to select for that plasmid, all right? And we'll talk about that more in the next. All right, so bacterial transformation, right? This is again the ability for bacteria to pick up DNA from the environment, right? And in a lab, we've kind of hijacked that ability. So the way that we do this is we have taken a target piece of DNA, for example, the cystic fibrosis gene from humans. We cut it out, right? At the same time, we cut a plasmid with the same enzyme, right? Cut the plasmid with the same enzyme. And we have put them together to make our recombinant DNA, right? So our recombinant DNA is going to have plasmid DNA and our target gene. We are going to take that target gene, that target DNA on the plasmid, right? So that's here. And we're going to mix it with bacteria. Usually the bacteria we're using is E. coli, right? And it's not the disease-causing E. coli. It's a different kind of laboratory strain E. coli. You heat shock it, and that then allows a very small percent of the bacteria to pick up the plasmid, all right? A small percent of the bacteria are going to pick up that plasmid. Those are now considered transformed. They've picked up DNA from their environment. Only a very small percent do this. Now, in order to select which bacteria picked up that plasmid, we are going to grow those bacteria on antibiotic plates. All right, so you may have seen these plates. They're called, they grow on LB agar, which is like a bacterial growth medium, and they're kind of like almost like a really hard jello. All right. And so if we have an antibiotic on that plate, only the bacteria with that plasmid, which has, remember, that antibiotic gene on it, antibiotic resistance gene, then only those bacteria with the correct plasmid that has the antibiotic resistance gene will grow on that plate. We can then take that bacteria, grow it up in a lab, and we could study the DNA, or we could transcribe and translate that DNA in order to study the protein in the lab. All right. So when we talk about gene cloning, right, this is a term that gets thrown out all the time, and I want to be really clear on what that actually means, right? So what we've done is, this is going to be an example of gene cloning. So here's an example of gene cloning. One, we're going to cut the gene of interest out of an organism. All right. So for example, our gene of interest is from a human cell. We are going to isolate the gene responsible for causing cystic fibrosis. We're going to take patients from patient DNA that has cystic fibrosis. We are going to look at that gene of interest. We are going to cut that gene of interest. And in the bacteria, we are at the same time going to cut a plasmid with the same enzyme. We're going to use DNA ligase to create our recombinant DNA, our plasmid, right? This recombinant DNA has our plasmid with all those features, like an origin of replication, a promoter, our target gene, which is shown here, and an antibiotic resistance gene, right? We're going to glue them together with ligase, and we're going to do a bacterial transformation to get that plasmid into a bacteria. Right? So now we have our recombinant bacteria, which it contains our plasmid with our gene of interest. Right? And now what we're going to do is select for that bacteria on antibiotic specific plates. And when we do that, the bacteria is going to grow on that agar plate that has antibiotics on it. This is gene cloning. So what we've done is we've had the bacteria grow and divide and make clones of our recombinant DNA for us, right? So the bacteria is growing and dividing, making many, many copies. Those are gene clones. Right? And then in the lab, we can do various kind of basic research to study either the gene or the protein. All right. So again, sometimes it helps to have like different examples of how different pictures, sometimes they click with students in different ways. So first thing, we are going to create recombinant DNA. We are going to cut the target gene. We are going to cut the plasmid, both with the same restriction enzymes. Those sticky ends will be complementary. We will then use DNA ligase to create our recombinant plasmid. Remember, plasmids are going to have an origin of replication. They are going to have a promoter. They're going to have our target gene, and they're going to have some sort of antibiotic resistance gene. Right? Second, now we are going to transform that plasmid into bacteria. So we're going to take our recombinant plasmid, we are going to put them with bacteria, and we are going to heat shock those bacteria. Only a small percentage of those bacteria are going to pick up the plasmid. We can select for those bacteria by growing them on an antibiotic. So we're going to place the bacteria on an antibiotic plate. bacteria without the plasmid will die. Bacteria with the plasmid will grow and make clones of our recombinant DNA. Right? We can then grow up lots of that plasmid. So we can take a little colony, they're called colonies, of bacteria, grow it up, and then we can study either the gene, like the DNA, or we can study the protein. We can have that in the lab, we can make protein. We can transcribe and translate that plasmid to make the gene of interest or make the protein of interest, all right? And next time in class, I'll give you lots of examples of cool transgenic animals, which have been made by recombinant DNA. All right, and we will stop there for now. Toodles poodles, bye, ciao.